remember that far back. So
It's there.
I
glad someone's being
decided to
honest.
just diagram on the whiteboards in the other building instead of show the PowerPoints. So since I know that you all studied diligently over spring break, I thought I would review this material. And by showing you the slides and then bring us up to where we are today. We actually are going to review the material today in the sense that we're going to go back to the structures of the nephron and revisit each of them as we talk about their role in the formation of filtrate. So, do you recall, you don't, that's why I'm doing this. We have the kidney-shaped kidney, and we have the cortex, and then we have the renal pyramid. So what I didn't really mention in lectures, we have renal columns in between the pyramids, and that's the left I talked about them. Okay, so I'm not going to test you on those or anything. Essentially, the pyramids make up the medulla. And filtrate is formed in the cortex. It is concentrated in the medullary pyramids. And by the time it leaves the pyramids through the papillae and enters the minor calyx, at that time we call it urine. Everywhere else in the nephron and collecting ducts, it's known as filtrate. Minor calyx is where we start with our transitional epithelium. All right, so anything that has urine in it is going to have a transitional epithelium, the multi-layered epithelium, not just the bladder, which is essentially what we learned in Bio 430. So the minor calyces join together to form a major calyx. This is just looking at the tip of a pyramid, papillae through a pyramid straight on. That's what that's showing there. And the major calyces join to form the pelvis of the ureter. The very last thing we looked at in lecture was the blood supply. So we have a preparation of a kidney like that up here, where the blood vessels were filled with a liquid plastic and then the tissue is digested away because obviously it's a sheep kidney. Obviously the kidney is dependent on the blood supply. We also have plastic-sized kidneys that have been triple injected. So you can see the structure here. This is why I don't do a dissection of a kidney, because it doesn't look any different than this. And the dissected kidneys stink of urine. It's all like an old baby's diaper. So you can see everything in this preparation right there. We have several. So we have the renal artery entering into the kidney. It breaks down into several segments. And then as it passes up between each pyramid through the renal columns, it's identified, remember, as the interlobar. Kidneys develop in lobes. And in the human kidney, those lobes, and sheep kidneys, those lobes smooth out. And the pig kidney is very bumpy, because each lobe, each pyramid has its own little bump on the outside. But those are the lobes are formed by the pyramids. And then at the, here's a model that we have. So here would be the interlobar arteries. Connecting each lobar artery, remember we have the arcuate arteries. And then branching out from the arcuate arteries at right angles, we have the interlobular, or since they're radiating out into the cortex, you may call them the cortical radius.
So the interlobular and the
Cortical radius are the same. Just some people refer cortical radius because it's confusing between interlobar and interlobular. So it's just kind of up to you. And then we don't name these branches out here at the very end. Okay. So when we look at, let's come back to the, we'll look at it here. When we come back to the blood supply, here we have, this would be a renal pyramid, interlobar artery, and here we have the arcuate and the interlobular, or cortical radius. I'm just going to say interlobular, okay? I don't have to repeat myself every time, both of them. Branching off of those are the afferent arterioles. And the afferent arterioles, remember arterioles are 1 to 2 layers of smooth muscle that provides the ability to constrict or dilate. And then the afferent arterioles lose their smooth muscle and become capillaries, and that first capillary band is identified as the glomerulus. It reforms into arterioles by forming a single vessel, regaining 1 to 2 layers of smooth muscle, and that structure is the efferent arteriole. The efferent arteriole, depending on where it is, if it's in a cortical nephron, the efferent arteriole here will form peritubular capillaries. So peritubular capillaries, and let me move forward here. Peritubular capillaries are always in the cortex. So they're typically formed by our cortical nephrons, and the cortical nephrons can have a small dip into the medulla, or the entire nephron would be in the cortex. So here we have the interlobular, the afferent arteriole, the glomerulus as a noun, or glomerular capillary, those are interchangeable terms, and then reform as a more narrow efferent arteriole. And the purpose of the peritubular capillaries, which is our cap bed 2, remember the glomerular are the cap bed 1, the peritubulars are the cap bed 2, is to selectively, well, not quite as selectively as you might think, but they reabsorb what was filtered in across the glomerular capillaries. And then the peritubular capillaries would enter into the interlobular veins and back to the arcuate veins and so on. I ignore the veins, because they have the same name as the arteries and the reverse flow. Now here is a juxtamedullary nephron, whose nephron loop is dipping down into the medulla. They have a unique purpose that we'll discuss today. So here the efferent arteriole is forming the vasa recta. Okay? Now not necessarily all of the efferent arterioles form vasa recta, some can form peritubular capillaries. But essentially most of our vasa recta are going to develop from the efferent arteriole. The ascending limb of the vasa recta can either return to the interlobular vein or it can enter into the arcuate vein. Okay, we did generally have more variations in what our veins do. So here the image is showing it return to the interlobular vein, but it can come into the arcuate vein. All right, so these are structures. That's kind of where we ended with our blood supply on Friday. And these are structures that we find only in the cortex of the kidney, only in that outer strip. So for the blood supply, only the glomerular capillaries are found there. Obviously, only the afferent and efferent arterioles are found there. For the nephron, we never find the capsule in the medulla. So if you're looking at a histology slide and you see these capsular structures, that's an easy way to tell that you're in the cortex. Okay. Vasa recta are only found in the medulla, and the papillary ducts, which are way down at the bottom, obviously are only going to be found in the medulla. We can find the nephron loop and the collecting duct in either location. So for our cortical nephrons, the nephron loop would be in the cortex. The collecting duct begins in the cortex and continues into the medulla. All right, so those are structures that overlap or are found in both places. All right, so now let's revisit these structures, looking at function. Remember I drew that long tube on the board and said the nephron is really just a tube with a balloon-like structure at one end. And so 3 events occur in the formation of urine. Actually, probably should change that to filtrate formation. It has to be concentrated before we actually form the urine. But all the contents of the filtrate and all the contents of the urine come from the blood. So the process of obtaining those contents is filtration. Okay? And that is the sole function of the Bowman's capsule containing the filtrate. You may see a term, the renal corpuscle, that is the capsule and the glomerulus together. I don't use that term because corpuscle is a cell. And this obviously is a lot more than one cell. But if you read it, that's the term they're referring to, those two structures together. Okay. Once the content is in the nephron tube, it's known as filtrate, and then we start to modify it. When we take material, remember I talked the story about the kids' stuff in the box? So kids are missing some of their toys, they go out to the garage and get their toys from the box. That is reabsorption. Okay? So that's taking material that's already in the filtrate, whether it's water or salts or amino acids, and returning it to our second capillary bed, the peritubular capillary. That's reabsorption. If we add material from the blood, so not only is it filtered, but we're adding even more, that's secretion. Okay. We typically do that with hydrogen ions when we're trying to buffer pH, with drugs that have been broken down the liver, so antibiotics or other drug metabolites will be added to the filtrate. So those three steps are what we're going to follow in your information. The content, what we determine is the final content. Not the amount, but content. All right? And then finally, the collecting duct is where we actually do filtration. So we don't, concentration. We don't concentrate the urine in the nephron. We use the nephron to create the ability to concentrate the urine, but we don't actually concentrate it in the nephron. So the first process we're going to look at is filtration. And if you recall, filtration is separation by size. In the kidney, we also add charge. But not only do we use pores of a certain dimension, but we use the presence of negative charges on some of the membranes or in the basement membrane to affect that filtration. But filtration as a definition is separation by particle size. It requires a pressure, and so the force is hydrostatic pressure. So of course here we're talking about blood pressure. There are opposing forces, all right, that affect how much of the hydrostatic pressure becomes effective, but that is the force that is essentially pushing these small particles across the filtration membrane. So the diagram that you're seeing here, this is the Bowman's capsule or nephron capsule. Remember, it is essentially a balloon that's formed of simple squamous epithelium. I'm going to erase this and redraw part of it in just a moment. Or I'll redraw it over here. And then in response to the tuft of capillaries that pushes against it, that bulges inward, and the inner portion of the simple squamous epithelium becomes intimately associated with the capillaries. So the capillaries essentially get wrapped around by the simple squamous epithelium as the capillary bed bulges inward. So this is a simple squamous epithelium. It's known as the parietal layer. Of the Bowman's capsule. And the epithelium that is intimately associated with the capillaries is known as the visceral layer. And then this is known as the capsular space. very similar to visceral pleura, parietal pleura, and pleural cavity, except the capsular space doesn't contain serous fluid. This is going to contain the filtrate. So once the plasma contents the fluid cross and enter the fluid of this space is filtrate. But as you can see here, the visceral layer is very specialized. It's not our typical simple squamous epithelium. It is still simple squamous epithelium, but it's not typical. Here's a histology slide. So here is the parietal layer, simple squamous epithelium. And you can see some blood cells in here. So that would be the capillary, but there's such an intimate association between the simple squamous of the visceral layer and the simple squamous of the capillary that it's otherwise difficult to separate the two. But this is the structure that when you see it, you know that you are in the cortex of the kidney. Right? Because we only have capsules and glomeruli in the cortex of the kidney. So let's take a closer look at the epithelia that are involved. This is showing just the blood vessels. The epithelia has been dissolved. So these are the tufts of capillaries that are associated with it. But let's look at the blood vessels themselves. The cells themselves. And we get it, when we look at it in the electron microscope image, we get a picture that looks like this. It's not quite complete, but this fluid, where you see the dissolved proteins, is the plasma of blood. This fluid up here is the filtrate. So we have a sandwich that is formed. Remember the last time we talked about a sandwich? Lungs. We had our respiratory membranes. We had the bread was 2 layers of simple squamous epithelia, and the filling was a little bit of connective tissue. Here we have even less connective tissue. The filling essentially is the basement membrane of the 2 epithelia. So this is the epithelium of the capillary, its basement membrane, which has a light and a dark layer. The epithelium of the visceral layer of the capsule, and its basement membrane, which has a light and a dark area. The 2 dark areas become fused. And this essentially is known now as our filtration membrane, formed of this sandwich. Right? So I'm going to erase what's on the board and diagram this filtration sandwich. Which essentially is something that looks like this. Okay? So we're going to have the plasma on the bottom. Filtrate up here. And our simple squamous cell, a little bit higher. So this is the simple squamous epithelium of our visceral layer of the capsule. But notice, instead of being just a flat cell, it forms these foot-like processes. Another cell, I'll draw it in a different color, overlaps and its foot-like processes are found in between and interdigitate with those. And a different cell would have its foot-like processes interdigitating here. So we have these slits, and that's one of 3 components that helps to filter. These cells are identified as podocytes. They form the visceral layer of the Bowman's capsule. The spaces in between are identified as filtration slits. And there's actually a slit membrane found in between each of these. There are specialized proteins and negative charges on this slit membrane that you can barely see here in between those, that slit membrane. Each individual foot process is identified as a pedicel. The foot processes of the podocytes are the pedicels. All right, so we go back to the scanning electron micrograph. This would be the podocyte, and these are the pedicels. The foot process is interdigitated. So this up here would be the capsule cavity, the capsular cavity, where the filtrate is located. Okay. Then we have the basement membrane. And this is the fused basement membrane of both epithelial cells. And then we have the capillary, simple squamous epithelium. It doesn't have foot processes, but it has pores or fenestra. Remember when we talked about the different types of capillaries? We talked about fenestrated capillaries? All right, so this is a fenestrated capillary. If you were to take a hole punch and punch holes in the plasma membrane, but when you punch the hole, it fused the edges so that when you look at the cell on the surface, it has divots in it that go all the way through. And think of an air mattress that you would use in a pool that has channels through so the water can come up and keep you cool, but the air is contained within the mattress because those channels have fused walls. So that's what these are like. And so these pores also contribute, of course, to the filtration and limit the size of what can pass through, but they're not the main determinant. More limiting are the charges in the basement membrane and the slit diaphragms. The larger things can pass through the fenestra or pores than can actually get across the basement membrane or the slit diaphragms. So this is the capillary. Simple squamous epithelium, and these are the fenestra. So here would be some views. These are the pedicels. So here's a slit diaphragm here, here, here. And down here as well, you can barely see the slit diaphragm. Let's see if I have another. You can see pores here and here. So the plasma tends to have a grainy look. If you're looking at the dissolved proteins, filtrate tends to be more clear. I think you have some images in the study guide in your lab packet for identifying the different cells. So in this lower power view here, this would be a photocyte. And here would be the pedicels, and you can even see the fenestra here a little bit better. Let me move this image up from the bottom. All right, so here are the slits, and here are the fenestra. So this is the process of separation by size. What are the molecules that can cross? Ions. Okay, so we have sodium ions, hydrogen ions, chloride ions, magnesium ions, water, amino acids, but not proteins, and glucose. Urea, that's the whole purpose here. Ammonia ions, or just NH3. Phosphate ions. Pardon? Potassium ions, definitely. All of our ions are going to be in here. Okay? There can be occasionally small proteins, but they're not a normal component of our filtrate or our urine. And if there are proteins in the filtrate or in the urine, it's usually due to damage of this filtration membrane. So high blood pressure can cause a damage to the basement membrane or to the slit diaphragms. Glomerulonephritis is a condition which, in response usually to streptococcus or staphylococcus, antibodies glutinate these bacteria and kind of plug up the antibody-antigen components, kind of plug up this filtration membrane, and that causes other areas of the membrane to be under higher pressure and can cause damage to segments, kind of blowing open the membrane, if you will, allowing large proteins to get into the filtrate. Yes?
That's a question about the picture on B. Is that just showing the bottom of the photocytes?
Yes.
A little bit.
So in the section, you're not seeing it. They would be connected above. They're just kind of, the angle of the section is not showing the photocyte itself as it is and showing it in the upper picture. All right, so that brings us now to a little bit of understanding of the pressures that are involved. So remember I said that we have the force. We were using a filtration paper and a funnel, and we were just pouring some mixture of a solution into it. We would have gravity would be the pressure, or you can attach it to a vacuum if you were doing a chemistry lab and increase the force that way. But our first primary force is blood pressure, right? So pushing the fluid and the particles against the capillary membrane is blood pressure. And that's what's shown here as the glomerular capillary pressure. And this is going to, of course, be affected by systemic blood pressure. So if someone has high blood pressure, they're going to have higher glomerular capillary pressure. If they have low blood pressure, low blood volume, they're dehydrated, trapped and haven't had water for a few days, they're going to have low blood volume and low glomerular capillary pressure. That can be regulated, of course, by changing the diameter of the afferent and efferent arterioles. So causing a constriction or dilation of those can affect it. Now, as that causes fluid and particles to move into our filtrate, this volume here is going to increase. And it's going to have an opposing pressure. So as we increase the volume here, it's going to have an opposing pressure, and that's the capsule pressure. Okay? Just as you blow up a balloon, the more air you put in the balloon, the more it's going to resist additional air coming into it. And we have another opposing pressure in the blood. Remember when we talked about the lymphatic system? What was the pressure opposing the hydrostatic force causing fluid to leave the capillaries? What was a major protein that had a pulling back force?
Albumin?
Yes, albumin. So obviously our large proteins are not going to be able to leave the blood. And so the large proteins in the blood are going to have an osmotic pressure pulling the fluid back. So this is an opposing pressure, okay? Now we don't have a similar osmotic pressure on the opposite side because remember our proteins are too large to move to the opposite side. So we don't have an opposing osmotic force. So typically our hydrostatic pressure in the capillaries is approximately 50 millimeters of mercury. And it's higher here than it is in typical systemic pressures. Our capillary pressures are typically not this high in systemic capillaries. Bless you. The capsular pressure is approximately 30 millimeters of mercury. And the osmotic pressure is approximately 10 millimeters of mercury. So we have a net filtration pressure typically of 10 millimeters of the 50 that we started out with. All right, we typically have a net filtration pressure of 10 millimeters. Now, there's obviously things that can change this. Okay? So let's say that the usual blood volume, anything that lowers or raises glomerular capillary pressure is going to lower or raise our net filtration pressure. Okay? So that makes sense. Anything that changes blood pressure here is going to change our net filtration. If we have somebody that has a liver problem, all right, and isn't producing sufficient proteins, we already knew that this could cause increased edema because we lack the opposing force in the capillaries. And we talked about the ascites, individuals who don't have enough protein in their diet, starving people, kids, people that have trouble processing that, lose additional fluid. So with that lower opposing force, we would have a greater filtration force. What about capsular pressure? Anything that blocks the outflow of our filtrate, so a kidney stone, blocking the ureter, it's going to cause the urine to back up into the ureter and back up into the minor and major calyces and back up into the collecting ducts. And so that can eventually back up here and increase this, which would decrease our overall filtration pressure. Okay, so it's a balancing equation on either side. Yes?
So when you increase capillary pressure, the capsule pressure increases with it?
So when you increase capillary pressure, we increase this number. This value here will increase, causing more filtration.
Okay.
It should only increase this marginally because it's going to increase the flow rate. So as we make more filtrate, that pressure is going to push it through the nephrons more rapidly. But if we block the flow rate, so if we go back to this image, well, that was on my other screen. If we go back to so if we have a blockage here with a kidney stone or your ureter stone, then this fluid backing up would eventually back up into the nephron, increasing the capsular pressure. All right, so let's take a look at how we regulate this pressure. We'll do a little bit now, and then after we talk about the role of the convoluted tubule, we can do that as well. So you recognize the diagram here would be our parietal layer of the capsule. Here would be our simple squamous membrane with the podocytes outside, our afferent arteriole, and our efferent arteriole. Now, I don't know if you recall, but when I was diagramming the nephron, after the nephron loop, I said that it came back up close to the capsule and the tubule passed between the two arterioles. That some artists took a license and had the loop go the opposite direction, but it's supposed to come back up, pass between the afferent and efferent, and then it becomes convoluted after that, all right? So as this distal part of the nephron passes through, instead of just simple cuboidal cells, like the tubule is normally formed of, as it passes close to the capsule, those facing the capsule become very tall and narrow. And the nuclei become very close together. And so this group of cells here is known as the macula densa, which means dense spot. Okay? That's part of a group of cells that are involved in controlling filtration pressure. Another group of cells are smooth muscle cells found in the afferent, in the wall of the afferent arteriole. These cells contain an enzyme. May I erase the board? Guys, contain an enzyme known as renin, which is involved in activating another protein. And then we have those gray cells that are called extramesangial cells. They affect the constriction, the diameter of the capillaries and effect of the podocytes. Okay? So these group of components are identified as the juxtaglomerular apparatus. Juxta, again, meaning next to the glomerulus. So the juxtaglomerular apparatus is involved in a local control of blood pressure for filtration.
Can you repeat that?
I'll write it on the board. Primary components are the juxtaglomerular cells, also known as granular cells. These are the smooth muscle cells of the afferent arteriole. And they secrete a chemical, an enzyme, known as renin. This is not the renin that's obtained from calf stomachs to curdle milk in the making of cottage cheese or something like that. It has two ends. And so this is an enzyme that takes, we'll come back and talk about, we talked about this very briefly And angiotensinogen is a liver protein, meaning it's produced by the liver. And it is activated under the influence of renin to form a molecule known as angiotensin 1. Angiotensin 1 is converted to angiotensin 2 in the lungs. Okay? So that's one part. We'll come back and talk about that more today. That's one part of our juxtaglomerular apparatus. Another component is the macula densa.
Can I ask a question?
Uh-huh.
Are granular cells just another name for
Yeah.
juxtaglomerular?
Yeah,
Okay.
you can use either term. These are simple columnar cells. Of the distal tubule. Not quite convoluted yet. So here is the afferent arteriole. These little pink dots that you see are the granules of renin that are in the juxtaglomerular cells. And this would be the blood flow through here. These are the macula densa cells. The Lacus cells, or poikasong, meaning pincushion. I'll put that star in parentheses because it's not always included in the juxtaglomerular apparatus. These are the extra mesangial cells, or I think it's poikasong or Lacus. So here would be the juxtaglomerular cells, okay, secreting the renin. They are sensitive to the sympathetic nervous system, so they have beta receptors. And alpha receptors. Here's the macula densa. And they have receptors that are osmotic receptors. That are sensitive to sodium ion concentration. Okay. It's kind of backwards, the way when you think of it. So if glomerular pressure is low, how would you relate that to the filtration pressure is low? How would you relate that to the flow of filtrate through the nephron?
It would decrease it.
It would be slow, okay? If filtration pressure is high, the flow rate through the nephron would be rapid. All right, going back. We have a slow flow rate through the nephron. Let's go back to this image here for just a moment to help you visualize that. So if we have a slow flow rate, are we going to have more sodium ions being resorbed or less sodium ions being resorbed?
More.
More. So when the tubule gets back up here, it's going to have a lower number of sodium ions in it. If the flow rate is rapid, there's not going to be as much time for sodium to be reabsorbed, so our sodium ion concentration is going to be higher when it gets back to the macula densa. Okay? So the macula densa cells are sensitive to the concentration of sodium ions. If sodium ions are low, meaning filtration flow rate is slow, then it sends a signal for the juxtaglomerular cells to release renin. Renin is going to activate angiotensin 1 to angiotensin 2, or angiotensinogen to angiotensin 1, which travels to the lungs and is activated to angiotensin 2. And there it has a couple of different effects, all right? Angiotensin 2 itself, as you can see by its name, is a vasoconstrictor. So this is going to result in systemic vasoconstriction. What's that going to do to overall blood pressure?
It's gonna increase it.
It's going to increase blood pressure. And it's also going to stimulate the release of aldosterone from the adrenal glands, which is going to increase blood volume by increasing sodium ion reabsorption. So that's the role of the macula densa, okay? Is just to sense, to detect the number of sodium ions that are present and then affect the role of the afferent arterioles. Okay, this is just another diagram of what's going on. I don't think it shows us anything extra here. Right. So glomerular filtration rate, this autoregulation, can occur just at the afferent or efferent arterioles. So just on pressure itself, okay? So if there's a large volume of blood pressure in the afferent arterioles, remember our Frank-Starling law? We stretch out the ventricles of the heart, they contract with a greater force. That's kind of what's going on here. We have a lot of blood volume entering the afferent arterioles where blood pressure in the glomeruli might be too high, and we overstretch the glomerular afferent arterioles. They react by constricting, okay? And then if the pressure is really low, they dilate to allow a greater amount of flow. So that's the myogenic, that means muscle originating. So that's a reflex in response to the the pressure changes in the arteriole itself. Now, with the tubuloglomerular effects that we're talking about here, so these, in addition to changes in pressure, the smooth muscle cells can release renin, or they can be stimulated to release renin because of low sodium, right? So the juxtaglomerular or granular cells will release renin in response to low pressure in the afferent arterioles or in response to signals from the macula densa, the adenosine molecules released from the macula densa can trigger that to occur, or through sympathetic innervation. So these are just 3 different events that can trigger the juxtaglomerular cells to secrete renin. So this can occur due to decreased blood pressure in the afferent arterioles, decreased sodium ions in the macula densa, causes the macula densa to send a signal, or sympathetic activity. Those are the 1, 2, 3 things that can trigger the juxtaglomerular cells to secrete renin. Here's another view. You recognize the capsule now, all right? Here's the macula densa. See why it's called a macula densa? Because the nuclei make this dense spot. So when you're looking at the microscope slides today, you can try to find those regions. So this is just a review of what we just described. And then the intraglomerular mesangial cells, these are inside, wrapped around the podocytes. So in between the podocytes, we have these intraglomerular mesangial cells. And when they contract, they function to decrease the filtration rate because they decrease the surface. So they make less available slits. Okay, decrease the number of effective filtration slits. So those are the intraglomerular, not the extraglomerular. So this is a summary of effects of decreased to low blood pressure, causes a decrease in filtration rate, which results in low filtrate flow because we have low volume. Okay, that allows more sodium ions to be reabsorbed across the filter from the filtrate across the nephron. The macula densa detect that, signal the afferent arterial juxtaglomerular cells to release renin. So renin release converts angiotensinogen to angiotensin 1 and then angiotensin 2 in the lungs. That itself causes vasoconstriction and also causes aldosterone to be released. So we end up increasing blood pressure systemically, which will increase glomerular filtration rate. All right. So we have our capsule, and we have a force which pushes the small particles across that capsule. We can regulate that force by changing the diameter of the efferent or the afferent arterioles, or we can regulate that force by changing the overall volume of blood that's coming in, okay, affecting systemic blood pressure. So now we have all the particles in our filtrate, and the next structure that it's going to pass through from our capsule is the proximal convoluted tubule. So to remember, our epithelium of our glomerulus is simple squamous, and now we switch to simple cuboidal. We're going to alternate between these two epithelia as we move through the nephron. Simple squamous and simple cuboidal, and back to simple squamous and back to simple cuboidal. So the proximal convoluted tubule is a simple cuboidal epithelium. Let's see if I have an image of it here I can show you. So these cross sections here and here, here, here and here, here, those are all proximal convoluted tubules. If you look, you can see the round nucleus, but you also notice how irregular and messy the lumen is. These cells have such long microvilli, not cilia, but microvilli. It almost looks like you took tissue paper and tore it. They're so very long, and in the section they tend to get clumped together. Okay? That is for the purpose, obviously, because microvilli are not mobile, that is for the purpose of increasing surface area. Because all of our amino acids, all of our glucose, as long as you're not a diabetic and have high glucose levels, and 80% of sodium in water is reabsorbed across the proximal convoluted tubule. This is not done under control of hormones, but is done under just concentration. So if we have a lot of glucose is reabsorbed. If we have a lot of sodium ions are reabsorbed. Okay? We have a lot of amino acids are reabsorbed. So it's purely numbers driven. We don't modify this by using hormones. That modification comes at the end of our nephron at the distal tubule, the distal convoluted tubule. Now erase the board. All right, so we have, I'm going to diagram a couple of simple cuboidal epithelial cells. occurs. We're going to have the filtrate here. And blood over here. What would be the name of this vessel? No? What's our cap bed to exactly? The peritubular capillaries. So we're talking about a simple squamous epithelia. Facing the filtrate, we have our microvilli. So all of these components have been filtered across our filtration membrane, through the pores and across the basement membrane and in between the filtration slits. All right? So setting this up, we're going to start at the basolateral membrane. So this is identified as the basolateral membrane. And that surface area is increased by folds. We don't have the microvilli, but we do increase the surface area with folding. This membrane is the apical membrane. So it has microvilli. And this has folds. So we're going to start here with the sodium-potassium ATP pump. Sodium is going to be pumped out. While potassium is pumped in. And that requires energy. So in this area, we have tons and tons and tons, well, not literally. It's packed with mitochondria. So if you were to look at an electron micrograph view, you would see a lot of mitochondria at the basolateral membrane. This is actually a question in our Bio 430 test last semester because of all the ATP requirements for our pump. All right, so that's going to move sodium out, and the potassium leaks back out again through leaky channels. And then we pump it back in again. This is active transport. So if we are actively transporting sodium out of our cell, what are we doing to the amount of sodium in the cell?
Decreasing.
We're lowering, okay? And we normally have a lower amount of sodium in the cell than we do outside the cell. So sodium wants to diffuse into the cell. And so it does. And it does so by going down its concentration gradient. It diffuses into the cell. But what have we done to that concentration gradient by pumping sodium out? We've increased it. So because there's less sodium in here now than there was before we turned on our pump, we've increased the drive for sodium to move down its concentration gradient, and it drags with it glucose. So this is still diffusion, because they're going from their high concentration to their low concentration. What kind of transport is occurring though?
Secondary active.
Secondary active transport. All right? This is active transport. This is our primary active transport. This is the one that requires energy. This is secondary active transport, which does not require energy. It's only occurring because this one does, but the secondary active transport itself does not. There's no ATP being used here. Okay? What do we call this type of transport when the molecules are traveling in the same direction?
Simple.
A symport. And when the molecules are traveling in the opposite direction?
Antiport.
Antiport. Okay. So, by the same mechanism, amino acids are transported across. So not just glucose. So glucose will do this. I'm going to put diffusion in parentheses because although it is moving down its concentration gradient, it does so faster. So typically secondary active transport is the first classification. So glucose does this, and amino acids are moved, are reabsorbed, and this is reabsorption because we're going from the filtrate to the blood. Now, as we move sodium out of the filtrate, water is dragged with it. So I don't know if we remember, sodium is our Mary, and water is her lamb. Wherever Mary goes, water is sure to follow. All right, so sodium is moved across, water is dragged with it, and water will cross two different locations. Water will cross through water channels known as aquaporins. That just means a water channel, okay? Or water, that's transcellular, going through the cell. Or water can go between. And this is paracellular. So most of the reabsorption, all the reabsorption of glucose, as much as the body can reabsorb, occurs in the proximal convoluted tubule. So if somebody has higher than normal levels of glucose in their blood, then the rest of the glucose gets stuck in the filtrate. It doesn't get reabsorbed. And that's where we get the name diabetes mellitus. Diabetes refers to an excess production of urine. So someone who is diabetic mellitus has polyuria as a sign. Anybody know what polyuria means? High volumes of urine. Why does that occur with diabetes mellitus? I just essentially gave you the answer here. Pardon?
Because you're not bringing sodium across, so water doesn't follow?
Change the molecule and you'll have it right.
Glucose?
Glucose. So the excess glucose has an osmotic effect. If it stays in the filtrate, water stays with it. So it has an osmotic effect, just like if you sprinkle sucrose on strawberries. It has an osmotic pulling effect of the water out of the strawberry. So it has an osmotic hold on water in the filtrate and keeps it from being reabsorbed back into the body, so increased urine volume. Okay. There's another, mellitus, by the way, means sweet. So with diabetes mellitus, we have increased urine volume. And if you stick your finger in the urine and taste it, it's going to be sweet. Okay, make a face, but urine is supposed to be sterile. Where does it come from?
Your kidneys?
Blood. Blood should be sterile. So we're not filtered. We don't have viruses and bacteria in our blood typically unless you have sepsis. And those would be, at least the bacteria would be too large to cross the filtration membrane anyway. And so it's only when it picks up, passes through the opening of the urethra, all right, and passes onto the surface of the penis or by the labia minora, that it's going to pick up bacteria. So if you were to take urine directly from the bladder, it should be sterile unless you had a urinary tract infection. It's going to have the waste products of nitrogen breakdown, the urea and the uric acid, all right, which when it smells like ammonia is gross, but you're not smelling waste, you're not smelling bacterial waste products like we do with fecal matter, okay? So diabetes insipidus means it has a bland taste. It's not sweet. And we'll talk about the cause of that on Thursday when we talk, or Friday when we talk about fluid, Thursday, when we talk about fluid balance. All right, let's stop here and take a break. When we come back, we will look at secretion and the function of the nephron loop. On Thursday, we'll talk about the function of the distal tubule, okay, where we have chemically controlled, hormonally controlled changes in absorption and secretion. So when we're done, when we come back talking about secretion and the nephron loop, lab will be the models, the structures of the kidneys that we've gone through already, histology, and you have your anatomy and histology quiz on Thursday. Okay? So secretion, remember, is in the reverse direction of reabsorption. So we're on page 22 of your lecture notes. And secretion is when we take material that may not, some of it was filtered, all right, but some of it wasn't. And so as it leaves the glomerulus and goes into the efferent arteriole, it ends up in the peritubular capillary. So we're still looking at the peritubular capillary here. And we add additional material to the filtrate, that's secretion. On Thursday, we'll talk about secretion of hydrogen ions. When we, well, actually Friday, when we talk about acid-base balance. We're going to come back to the proximal convoluted tubule and talk about how hydrogen ions are secreted. But quite a few things are secreted. They're listed on page 22. And remember, the purpose of kidney excretion is to get rid of nitrogenous waste. And urea and ammonium ions and creatinine are the most common. Uric acid is usually too solid. But we reabsorb most of the urea, and then we secrete urea. So the urea nitrogenous waste form that we have in the filtrate has ended up being secreted. This particular diagram shows a process of the formation of ammonia ions here. So glutamine is an amino acid. It can be filtered, right? And it can also be secreted. So what we're saying is not 100% of everything gets filtered. So some of it gets filtered, and the blood in the peritubular capillary would then have less of that than it had in the afferent arteriole. Does that make sense? So it comes into the glomerulus. Let's go back again to this image. We'll use this one. So it comes into the glomerulus. Let's say it has 100 molecules of amino acids. Some of those are going to get filtered. And when the blood goes out the efferent arteriole into the peritubular capillary, it's going to have less amino acids in the peritubular capillary than it did in the afferent arteriole. But in the proximal convoluted tubule, we reabsorb all of that, so by the time it gets back into the interlocular veins, it has the same amount of amino acids that it had to begin with. All right, that's essentially what happens with amino acids and with glucose. We filter it, and then we take it back in again. We reabsorb it. But sometimes the body wants to get rid of more material. So on Friday, when we talk about acid-base balance, and let's say the body has too much hydrogen ions, so it wants to secrete it. So hydrogen ions will be filtered into the proximal convoluted tubule. The peritubular capillaries will have less hydrogen ions than the afferent arteriole, but then they will secrete even more. So now the proximal convoluted tubule filtrate ends up with more, and the peritubular capillaries have even less than they did when they entered the efferent arteriole. All right, so it's a way of adding more tube. So we end up secreting the various drug products, so those are listed down at the bottom of page 22. And here are amino acids, some were filtered, here amino acids are secreted, and both of them end up being broken down to form ammonium ions and bicarbonate ions. And ammonia ions, this is already in your lecture notes, so I'm not going to re-diagram it. But notice here we have secondary active transport, but with an antiport. The secondary active transport that I showed you here with glucose and sodium were symports. Here, they're antiports, where the sodium is pulled into the cell via the primary active transport and exchanged for ammonium. So it's still secondary active transport, but in the opposite direction. So it doesn't have to be a symport, it can be an antiport. Right? And so this is a way of excreting additional nitrogenous waste products into the filtrate. So before we leave the proximal convoluted tubule today, we'll come back to it on Thursday and more on Friday. This is where we make the primary, the only changes to determine the content of what's in the filtrate. By the time the filtrate leaves the proximal convoluted tubule, all right, and enters our descending limb, what is actually going to be in the filtrate is already determined, not how much. So we will still remove sodium and add different components, but what is there is already determined. So we've taken out the glucose, we've taken out the amino acids, we don't add that back anymore, all right? We may secrete bicarbs or reabsorb hydrogen ions, so we're changing amounts of material, but not actual individual components. So there won't be any new item that's present in the filtrate that isn't here by the time we leave the proximal convoluted tubule, and there won't be anything absent. It may be less, but nothing absent. So we leave the proximal convoluted tubule, and again, that determination is based on numbers. We don't use hormones to increase or decrease the amount of material that is removed. If you've eaten a lot of sugar, then you're going to have a lot of glucose in your blood. A lot of glucose will be filtered. A lot of glucose will be reabsorbed. Okay? You've been on a starvation diet. You're not no low carbohydrates, but maybe a protein diet, carbs but high in protein. You're going to have a lot of broken down amino acids. Those may not all, most of them will be reabsorbed, but maybe not all. And so you might have a few amino acids still in your blood. We're not going to add, we're not going to change that and take them out later on. So whatever is flowing into the descending limb of our nephron loop is the same material, again, maybe more, maybe less than what we eventually see, but nothing new. Nothing removed completely, nothing added that isn't already there. So now let's move into the nephron loop. So this is looking on page 24. And we'll be done about 9:30 or so, okay? And then we'll pick up distal tubule with electrolyte and water fluid control on Thursday, and then acid base on Friday. So this is considered to be a cortical nephron, even though it has a short loop in the medulla. Other cortical nephrons don't have any loop. And this is a juxtamedullary nephron. This is the one that we're going to talk about, okay? The juxtamedullary nephron, with its nephron loop in the medulla, its role is to set up an ion gradient. It does not concentrate the filtrate. the purpose of the ion gradient is to concentrate the filtrate. That happens in the collecting duct. The concentration itself happens in the collecting duct. By the time the filtrate leaves the medulla in the nephron loop, it is actually hypoosmotic. So normal osmolality, you know what I mean when I talk about osmolality?
Osmotic pressure.
It relates to osmotic pressure. Osmolality is a reference to the number of particles that are in a solution. So if I take 10 molecules of glucose and 10 molecules of sodium ion and put them each in a beaker with 10 mils of water, which one would they have the same, or would one of them have a higher osmolality?
They have the same.
So here's our beaker, 10 mils of water. And I add 10 sodium chloride molecules to this one, and 10 glucose molecules to that one. All right? So here's 3, 4, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So same number of particles per volume. So at this stoppage point in time, they have the same osmolality. However, does sodium chloride stay in water as sodium chloride?
No.
No. This is going to dissociate to sodium ion and a chloride ion. So I'm going to actually have, instead of 10 particles, I'm going to have 20. This now has a higher osmolality. Okay? Glucose does not dissociate into smaller components. Because sodium does, it creates a higher osmolality. All right? So osmolality is a reference to the number of particles, and we do it in moles and so on, but it's simply in reference to the number of particles per given volume. So normal blood osmolality is 300 milliosmoles. So that's what we have in the blood, and that's what we have in the filtrate. So when we start our descending limb, we have 300 milliosmoles. By the time we leave the nephron loop, we have 100. So we obviously have not concentrated the filtrate. That is not the role of the nephron loop. What happened to those 200 milliosmoles? What happened to those particles of sodium? They are removed from the filtrate, and they are used to set up a gradient of particles that is more highly concentrated at the base of the loop, at the actual loop itself, than it is up here. That is the whole purpose of the juxtamedullary nephron loop, is to set up this gradient where we go from 300 milliosmoles in the extracellular fluid to a high of 1,200 to 1,400. Typically you'll say 1,200, but the maximum is about 1,400 milliosmoles. And we do that by a loop. So we have fluid flowing in this direction, fluid flowing in that direction against itself. You guys remember what we called that when we had this occurring with the arterial blood and venous blood in the testes? This is counter current flow. Now, we talked about the vasa recta briefly. And the vasa recta, as you can see on this diagram here, has a counter current flow to the nephron loop. So the nephron loop is flowing and the filtrate is flowing in this direction. Our vasa recta is flowing countercurrent to that. So the nephron loop has a countercurrent flow. And our vasa recta has a countercurrent flow to the nephron loop. However, we have pumps that are present in components of the nephron loop, in the thick ascending segment of the nephron loop. So not only is this a countercurrent flow, but because of the pumps, the nephron loop becomes a countercurrent multiplier. And so the rest of the lecture is to see how this happens. But before we move into that, I just want to reinforce the nephron loop, the filtrate in the nephron loop is not becoming concentrated. It's actually going from normal isoosmotic to hypoosmotic. It ends up being 100 milliosmoles instead of 300 milliosmoles because it's losing ions to this gradient. The gradient is found in the medullary pyramid. What else do we find in the medullary pyramid? The collecting ducts. So as the collecting duct passes down into the medulla, it has water channels, those aquaporins, and we're going to talk about this on Thursday. And these aquaporins are opened in the presence of a hormone. Anybody remember that hormone?
Antidiuretic?
Antidiuretic hormone. So in the presence of ADH, we insert these aquaporins. And now let's say that our filtrate is 100, coming from our ascending limb, and it passes in through radiant extracellular fluid. That is 300. Which way is the fluid going to be pulled? Out to our gradient. So now our filtrate becomes 300. It passes by the gradient, which is now 600. If we have additional aquaporins open here, fluid is going to leave, and now our filtrate is going to become 600. Okay? So it is in the collecting duct, with the presence of aquaporins, that we are able to concentrate the filtrate. Let's say you drank 8 cups of water this morning. Your body's not going to be making ADH. All right? You don't need to reabsorb all 8 cups of water. So we do not have these aquaporins present. Our filtrate comes in at 100. It leaves as 100. And you have a very dilute, almost colorless urine when you go to the bathroom. So all this concentration or not concentration takes place in the collecting duct. It would take a long time to set up our gradient and remove our gradient compared to adding or removing aquaporins. So the gradient is set up and maintained by the nephron loop in the vasa recta. If we need it, we insert aquaporins. If we don't insert aquaporins. And that's where the concentration takes place. So let's take a look at how this occurs. I'm going to match this up to my chart. I'm going to draw this in the opposite direction as I did over there. So descending limb, ascending limb. And where the thick segment is going to be variable depending on our location of the nephron. So The descending limb is permeable to water. It is also permeable to salt, but it's permeable to water. The ascending limb is impermeable to water. And especially in the thick segment, it has sodium active transport pumps. So it is pumping out sodium. Chloride happens to follow passively. These use ATP. And the pumps can maintain a difference of 200 milliosmoles between what is in the filtrate and what is outside the filtrate. All right? So if outside the filtrate we have 300 milliosmoles, inside the filtrate the maximum difference we can maintain is 100. If outside the filtrate we have 500 milliosmoles, the difference inside the filtrate that we can maintain is 300. We can't go 100 and 400. We don't have enough. That takes too much ATP. The cells aren't capable of maintaining that difference. So remember, the whole purpose here is to remove particles that we can add to the gradient. So let's go through a sequence of images. And I have these as a separate file on D2L. So this is the same thing that's shown in this image here, but I think it's easier to understand if we look at it one at a time. Okay. This comes from the textbook by Laura Sherwood that I made up a diagram to illustrate because I think it's a little bit clearer. So on the left side we have the descending limb, and on the right side we have the ascending limb. Okay? These we kind of allow the filtrate to flow in and fill the loop. We don't have any pumps turned on now. We just have a stationary system. The filtrate from the proximal convoluted tubule has flown into and up the loop. No pumps are on at this point. So our filtrate coming in is 300 milliosmoles, and our extracellular fluid in the medullary pyramid is 300 osmoles. We haven't done anything. Okay, this is just like it would have come in from the proximal convoluted tubule. So then we turn on, remember this is the ascending limb, so we turn on a sodium-potassium or a sodium pump, and we pump sodium ions out into the extracellular space. Remember, we're doing it on the ascending limb. The ascending limb is impermeable to water. So normally when we pump sodium out, water follows. Okay, that's not going to happen here because the ascending limb is impermeable to water. So we're only going to be losing sodium and chloride. So because we've lost ions, the osmolality of inside the filtrate becomes less and the osmolality outside the filtrate becomes more. That make sense? We're taking from here, we're taking 100 from here and adding it to the outside, into the extracellular fluid. So that makes the inside 200 and the extracellular fluid 400. Now remember, the descending limb is permeable to water. So this is a higher osmolality. There are aquaporins present here. There are water channels present here. And so it's going to pull the water out of the descending limb. And now that's not going to make it more concentrated, but it can make it as concentrated. What's the next question that should be in your mind right now? If we pull water out of here into here, shouldn't this become more dilute? Yes. That's the purpose of our vasorectum. soaks up that water and helps to maintain this at that concentration. Because yeah, it would just become dilute and we'd lose our gradient, okay? So now we turn on the pump up here, do the same thing, pull water out, and make that a higher osmolality. That's for our acid base, I don't know what ended up there. And so now you can see that the ascending limb is 200 milliosmoles lower in osmolality than the extracellular fluid, and that the descending limb is the same osmolality as its surrounding fluid because water can leak. Okay? Another thing, the ascending limb is becoming hypoosmotic. Right? Remember what we call this, not just a countercurrent flow, but a countercurrent multiplier? We go from 300 milliosmoles to 1200, which is way more than 2 times concentrate, 200 milliosmoles higher. All right, if we didn't have the countercurrent multiplier, this is the maximum we could make a concentration of the extracellular fluid. Now, we're going to turn on the flow, and we're going to move some of this 400 milliosmoles over to the ascending limb. Okay? So 200 milliosmoles is going to go into our distal tubule, more 300 is going to come in from our proximal tubule. So now we have 300 again, 400, but over here now we have 400. This is the function of the countercurrent multiplier. So now we pump out sodium ions from our ascending limb again, creating a 200 milliosmole maximum difference. You know where this is going. The 500 is going to pull fluid out of there, and we're going to make that 500. Again, maintaining a difference of, maximum difference of 200. Okay? So now we have a gradient of 150 to 300. So we pull that over, we move some of the 500 over, 150 goes up into the distal tubule, more fresh 300 comes down, turn on our pump. Always maintaining a gradient of 200 milliosmoles difference. And now you can see we're starting to create this gradient. Okay. This is becoming more concentrated, but by the time it leaves, it's actually less concentrated. Okay. And so when we're done, we have a gradient of our extracellular fluid in the medullary pyramid of up to 1,200 to 1,400 milliosmoles. By the time it actually leaves the distal tubule, though, it's hypoosmotic. So yes, it does get concentrated in the descending limb as part of our countercurrent multiplier, but the final filtrate is not concentrated when it leaves, it's actually diluted, because we've taken ions out to place in the extracellular fluid of the pyramid. Now, there is a species of rodent that lives in the desert called a kangaroo rat. It doesn't have very much moisture. It's dependent on dew at night. And it excretes its nitrogen process in almost a solid form of crystallized uric acid, so it takes every bit of moisture out. Its urine filter, its urine can be concentrated up to 4,000 milliosmoles. 4 times higher than our kidneys. Anatomically, the difference is they have longer nephron loopholes. Okay. So the only way we could increase the concentration of our filtrate is to anatomically, structurally change and create a longer loop so that we have more surface area for pumps for that counter multiplication. Okay. So a critical thing from here that I want you to be able to recognize is obviously the descending limb is permeable to water. That's how we increase its osmolality to allow the continued pumping of sodium ions out of the ascending limb, which is not permeable to water. So the ascending limb has the sodium pumps. The descending limb is permeable to water. Anytime, so the descending limb and the extracellular fluid are always going to have the same osmolality. The ascending limb is going to be 200, maximum 200 milliosmoles, lower than that at any given point. So that creates our gradient. So if we go back, and again, there's a separate file for these slides on D2L. That is the osmolality of what you excrete. Depending on the amount of ADH, it would reach a maximum of 1,200 to 1,400. Okay. And then the vasa recta flows in the opposite direction of the loop and in opposite direction to itself and picks up sodium and water because we're constantly replenishing this and refreshing it. But it's also going to pick up water from the collecting ducts. So as we do pull water out, that could also dilute our gradient. So the vasa recta are there to help to maintain. The gradient is formed by the nephron loop. The vasa recta help to maintain it through the reabsorption of the water and sodium. All right, so we'll stop here with lecture for today. On Thursday, we will look at the distal tubule and distal convoluted tubule because this is where we see hormonal control. This is where aldosterone is going to have its effect, and to some degree at the distal end of the collecting of the distal tubule, and then in the collecting duct, we have the role of ADH. So on Thursday, we'll look at the control of water, to degree a few minerals, all right, like calcium ions and so on. And then on Friday, we'll look at hydrogen and bicarbonate transport across as we control acid base. Lab today is going to be familiarizing yourself with the models and the histology. Let me see if I have. We have a couple of models that are a little bit scary. Let me just bring them out and show you. There's not actually a lot of different structures for the urinary system. So we have the overall anatomy of the kidney, the ureter, the bladder, and the muscles. Remember I talked about the fact that the muscles are supported by psoas major and quadratus lumborum. We listed those muscles on Friday. We have 3 of these sets that show the overall kidney with a view of the pyramids and the blood vessels and the calyces and the pelvis and ureter. This is showing the pyramid and the cortex, all right, and the peritubular capillaries and vasa recta. And then this is showing what we first started out with, our capsule, the afferent arterial, efferent arterial, the glomerulus, and the podocytes, the proximal convoluted tubule leading that. This is a newer one. We have an older one here. So this is representing one medullary pyramid with the collecting ducts. And up here, so you can see the podocytes, you can see the macula densa and the juxtaglomerular duct. So those structures have been opened up. And in the scary model. That's this one. It's plaster, so it's heavy. Same structures we've all looked at, so it's just the multiple colors that make it scary, all right? So you're already familiar with everything that's on here. So this is pyramid, this is cortex, okay? Running along the base of the pyramid, we have the arcuate, artery, and vein. So we're not seeing the interlobar, but here's the arcuate. That would make this the interlobular or cortical radiate. Coming off of it, we have the afferent arterial. And then coming out of the capsule, we have the efferent. And you can see here the efferent forms the peritubular capillaries in the cortical nephron. And here the efferent is forming the vasa recta in the juxtamedullary nephron. You don't have to worry about this right here, just the vasa recta. All right, so in identifying the parts of the nephron, you just have to look at your orientation. So coming out of the capsule, we have the proximal convoluted tubule. That's the bisque color, kind of a beige orange one. So that's all proximal convoluted tubule. Then you see it drop down. That's the descending limb of the nephron loop. So here's the descending limb of the nephron loop. Comes back up as the ascending limb. Becomes distal convoluted tubule and enters into the collecting duct. So if you see it going into the collecting duct, you know it's distal. If you see it coming off of the capsule, it's proximal. All right? So it's just one tube that has convolutions and loops, but you can trace that around to figure out where you are. So the multiple colors can be a little bit disconcerting at first, but you just trace it and know whether you're going away from or towards the capsule. You can figure out whether you're proximal or distal, descending or ascending. Okay? So that's that component. The only other part that we have model is this. We're showing the bladder and the ureters and so on. Well, we do have individual kidney models, which are found on here, but we have these individual kidney models that you can identify calyces and blood vessels on. So we don't have a lot of different parts like the respiratory system. Histology-wise, we go back and forth from simple squamous to simple cuboidal. Our nephron loop, the thick section is simple cuboidal, the thin section is simple squamous. We get back to the distal tubule and we're back to simple cuboidal again. So we alternate between those components. Alrighty, so on Thursday, lab is urinalysis, and we'll have some fake urine to test. I was going to buy the refractometers. It was going to end up costing me $280 of my own money. I decided not to do that. So I'm going to wait until I can get the department to purchase them. So I'm not going to have you guys drink a ton of water and eat a ton of salt and go to the bathroom every 30 minutes. Okay? You want to?
No, thank you.
Just wonder if you're being sarcastic or not. Because I would have to tell you today about which groups you're in and whatever to drink. So we will work with results as if you were to do that. Okay? So we'll go over that on Thursday. You'll be testing fake urine for different components and then looking at the results as if you were in a group that drank seawater or distilled water or caffeinated water or something like that. Okay? So then next year's group gets to be the guinea pigs. And if I have to buy the refractometers, if I buy one a month for the next year, it won't be so bad. They're $30 on eBay, but I wanted to get enough for each group, and I'm not sure. I need to check and make sure $30 is a good one. They have them for distilling, they have them for pickling, but the ones I wanted to get were veterinary ones, so that they were in the right range of what we needed to do. Alrighty, so I'll pull the slides out. You're just looking on the slides. You want to be able to identify them. Well, I have those up there. Let me show those to you. You want to be able to identify whether you're in the cortex or whether you're in the medulla. So again, if you see this, you're in the cortex. If you see proximal convoluted tubules, you're in the cortex. All right? In the nephron loop, you're going to see collecting ducts, which can be in both. And they can have simple cuboidal, they get the epithelium gets simple columnar as we move into the medulla. Now these structures right here are simple cuboidal, but notice that they don't have the tall microvilli. Their microvilli are sparse and very short. Those are the distal convoluted tubules, all right? And then we have simple squamous epithelium. So we have the thin segments of nephron loops there, okay? So you should be able to identify the capsule, you should be able to identify proximal convoluted tubules, distal convoluted tubules, and then I'll show you some slides once lab gets started of the medulla. So let me go grab some of those slides. The older ones, again, tend to be more contrast and more sharply stained. See if you can find some macula densa on this slide. Can anybody see one on this slide? It's not very prominent.
It's on the left then.
Right.
It's actually right here. Okay, but usually they're a little bit taller than that. So you want to look for this on like 10x, where you can do a quick scan to try to find those. So take a break, start whenever you want to. I'll pull some of the slides up, throw some slides up in 10 or 15 minutes or so, kind of show you some more images and what those look like.
Copies of stuff.